Hello, everyone, and thank you for joining us today. I'm Sean Fensky, Editor-in-Chief of Medical Product Outsourcing and moderator for today's program. You logged on to the MPO webinar, Requirements and Impact of the New Guidance, ISO 10993-23, Tests for Irritation. Before we get started, I have a couple of housekeeping announcements. For those of you who are interested in the information you hear today, once the webinar has ended, or if you'd like to forward it to a colleague, this session will be archived for a year. The archive will be able to be accessed through the Medical Product Outsourcing website, which is at mpomag.com. Also, we will be accepting questions throughout the presentation. You can type the question into the Q&A box in the webcast window. We will collect all questions and hold them until the end, but please feel free to type in questions as you have them. And now, before I introduce today's speakers, I'd like to take this opportunity to thank our sponsor, Nelson Labs, a Sotera Health company. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barriers industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. And now, let's get started. Today, we're joined by two members of the Nelson Labs team, Dr. Sophie Michel, biocompatibility expert, and Audrey Turley, senior biocompatibility expert. Dr. Michel got her PhD in biomedical engineering from the University in Maastricht in 2014. Thanks to her experience in chemistry and biocompatibility, she joined the team of study directors at Nelson Labs Europe in 2015. Where she focused on chemical characterization for medical devices as per ISO 10993-18. In 2019, she became part of the Nelson Labs consulting team that advises sponsors on all aspects of the ISO 10993 series. Since 2019, Dr. Michel has also been a member of the ISO 10993 Technical Committee. Audrey Turley has over 20 years of experience working in research, laboratory, and test design function in the medical device industry. She is a biocompatibility expert, having spent 15 years in the lab performing all the in vitro tests offered at Nelson Labs, which includes cytotoxicity, hemolysis, and genotoxicity. Audrey was responsible for the development of the in vitro mammalian chromosome aberration assay at Nelson Labs. She was also on the team at Nelson Labs, which developed and validated the in vitro irritation assay for medical devices. Now, beginning today's presentation will be Dr. Michel. Dr. Michel?
Thank you for this nice introduction, and good morning or good afternoon, everyone. Today, Audrey and me are going to speak to you about the new guideline ISO 10993-23 test for irritation. So I would expect that most of the people attending here today are already quite familiar, I would say, with medical device. And so you probably know that before, up to now, of January 2021, irritation was actually part of ISO 10993-10 together with skin sensitization. And beginning this year, this has changed, and irritation has now moved to its own part dedicated, which is ISO 10993-23 test for irritation. So ISO 10993-10 will stay active, but it's going to focus on skin sensitization only. So you will have one part, ISO 10993-10 for skin sensitization, and one part, ISO 10993-23 test for irritation. And of course, in this webinar today, we are going to speak and focus on ISO 10993-23. But first, let's step back and remember what is irritation. So irritation is defined in ISO 10993-23 as a localized non-specific inflammatory response to single, repeated, or continuous application of a substance material. And it's important to note that it's a reversible reaction, which is mainly characterized by local erythema and swelling. So there is nothing new in this definition because it was already identical in ISO 10993-10, but it's just to make sure that everybody understands what we are going to be speaking about today. And irritation is actually an endpoint of evaluation in the biological evaluation chart of medical device in ISO 10993-1. And you can see that the endpoint is here just below the blue arrow. If we look more closely, we can see that irritation is actually an endpoint of evaluation for every medical device. That means whichever the contacting time and whichever the contacting manner, you will need to evaluate for irritation. So it's actually an evaluation, so it's part of a risk assessment process. You need to understand how you're going to look for this endpoint. It can be that you will address it by information on your final medical device by collecting data on it, but it can also be that you will have to address it through testing. All this is actually part and defined in your biological evaluation plan. But of course, we have to face the fact that many medical devices will still have to go through testing. And of course, this used to be a little bit, I would say, problematic up to now or concerning, because in ISO 10993-10, the only way to assess for this endpoint through testing was actually by performing the test in animal. And so you can imagine that because this endpoint is actually applicable for every medical device and a big majority of them will have to go through testing, that actually involved a very large amount of animals. And for this purpose, the medical device was in most cases extracted in non-polar or polar solvent and then injected in the animal. And so there has been really big research going on how we can try to change this and go through in vitro methods. And this is actually what has been succeeded with this new ISO 10993-23 guideline, which deals with the fact that you need to consider in the in vitro method first. And of course, this method has been developed and validated for medical device. How do you do this? You, in most cases, extract your medical device again in a polar and apolar solvent, and then you inject this extract, you put this extract on epidermal skin tissue. So this is for the background, and now we are going to focus, of course, much more on ISO 10993-23, its content. And for this, we are going to go over the general principles which are outlined in the guideline. So ISO 10993-23 actually gives you different tools that you can use to assess the irritation potential of a device. So a first way to do it can be to go through a literature review as per ISO 10993-1. So you understand how your final medical device is made, what are the materials which are composing it, and you are going to evaluate if these materials are standard in the medical device industry, if there is already quite some literature on it, and of course if there is, for example, literature which is relevant to the assessment of irritation. Another way to do this would be to understand in great detail your manufacturing process, for example, and gather physicochemical information on your device. So here you want to understand what are, for example, the processing aids or the cleaning agents which are used, and see if they have any impact, if they can have any impact on the final irritation potential of your medical device. So that's the theory. What does that mean in practice? So let's imagine that you are putting on the market a silicone medical device. Silicone is naturally a material which is well assessed in medical history, and in biocompatibility you can find quite some literature already on this material and also describing the irritation response of this material. Let's imagine also that your raw material supplier has already been performing quite some biocompatibility tests on the raw material, for example, USB class 6, which already includes irritation as a biological endpoint. Then if your device is manufactured by injection molding and there is no processing aids used, and the packaging materials are quite well known, for example, Tyvek and polyethylene terephthalate, then maybe you will have enough information already to address the safety of your device towards irritation. And so you will be able to stop there and not proceed to testing because your assessment for irritation will be complete. So that's, of course, an example. It would highly depend on your medical device and the amount of information that we can collect and the impact of the manufacturing process. So it's really device-to-device dependent. But it's to give you an idea of how you can use this tool to assess the safety of your product. Another example would be you are exactly in the same situation than before, but you have carefully evaluated your manufacturing process and you have seen that there is in the manufacturing process a known irritant which is used. Then a way to assess irritation can also be to perform a targeted chemical analysis to define the concentration of this specific irritant and then make a toxicological analysis afterwards to determine if its concentration is safe to the patient. Another way could be to perform a cleaning validation to demonstrate that the irritant is effectively removed from the final product. And if you are in this situation, then again, it can be that you will not need to proceed through testing. So a third tool is naturally to proceed with, I would say, testing. And ISO 10993-23 asks you to consider in the first stage in vitro irritation. This is really clearly mentioned. In vitro irritation tests shall be performed before animal testing or human patch is considered. So that means that for every new medical device which is coming on the market, or for example for, I would say, already commercialized medical markets, but for which you would need to reassess irritation as part of a change, for example, or a risk assessment, then you would need to consider the in vitro irritation test first. There is also mentioned that there is some exceptions. For example, there is for special irritation tests relevant for medical device intended to be applied to a specific area, like the vaginal or eye epithelia, then the in vitro models are not adapted. So what does that mean in practice? That means that if your medical device is a surface medical device in contact with intact skin or with breach compromised surface, or if it's any kind of externally communicating device or any kind of implant medical device, then you will need to consider the in vitro irritation test first and proceed to this test. You can see that the mucosal membrane is circled differently. Here, this is actually this kind of contact where you would need, as part of your biological evaluation plan or risk assessment, to define if the in vitro model is adapted or if you have to consider the in vivo test directly. Another way to assess irritation is also to perform in vivo animal tests, but here this is only if needed and justified. Once again, what does that mean? This would be only authorized if your device is coming into contact with special tissue of mucosal membrane, for example, vaginal, penile, ocular, rectal, or oral buccal. Then here, indeed, the in vitro model is not adapted and you can directly proceed to in vivo irritation. finally, a last way that you can assess irritation is to proceed to clinical studies, but you can only proceed to in clinical studies if you have already assessed the irritation response of your device by the in vitro or the in vivo method. So naturally, you can see that actually the big novelty of this guideline is to propose an in vitro irritation test, which is validated and developed for medical device. And here I'm going to give the speech to my colleague Audrey, who developed, this method as part of being a member of the ISO committee on irritation, and so she really knows a great lot about this test.
Thank you, Sophie, and thank you to everyone who's joined us today. I know that there's been a lot of discussion in the industry about this method and this change in the standards, so we're really pleased to have you join us today for our contribution to this discussion. So I want to start off first by talking about the tissue. That's an important part of this system because I think there's a lot of confusion in the industry if this is really a complex enough system to assess irritation. So really what we want to start off with is looking at the tissue. So you can see that we have two examples side by side histopathology stained tissue, and the in vivo, so the live, from an actual donor, you can see that on the left. And then we see the in vitro tissue that's been grown up on a plate. This is from Skin Ethics, so this is from the lab in France. And you can see the differentiated tissue layers here, and that it's very similar to in vivo. So this is a component of a requirement for these tissues, that they are stained and analyzed to understand and verify that we are seeing tissue differentiation when the tissue is grown in vitro. So we get a great deal of, sorry, I went to the wrong slide. Let me go here. We get a lot of information from the tissues from OECD 439. So this is the standard that we used to guide this development method for devices, because this has already been a method used for cosmetics and chemicals. And so there's quite a bit of information and requirement on the tissues within this standard that is referenced in -23. So OECD 439 will list the tissues that have determined and been shown to be effective for for assessing the irritation potential of chemicals. This is their most recent updated list. And for the validation for -23, for the round robin study, we use the epiderm tissue and the Skin Ethic tissue. So these are from two different suppliers, because we just didn't want to use one source, right? We always want to use a couple different tissues to show the robustness of the method. And you'll see that in -23, there is a requirement that if a new tissue is to be used, which we definitely want to support having more tissues be used for this, that there has to be some type of study performed to show that it is as comparable as the tissues we used in the validation. And so that's one component in -23 that we specify. And also in 439, OECD 439, there's requirements for these tissues that have to be met so they can be used. So this is like their quality control. So we can see, I apologize, I didn't update this one table. So you can tell this table looks a little outdated. This doesn't contain the most up-to-date list of tissues. However, these limits for the tissues listed are the same. But one of the requirements for the tissues is they have to show a proper barrier function. So again, this is another step to show that the system is complex, that it's not just cytotoxicity or just exposing, just a monolayer of cells. This is a complex system that does function like skin with some of the properties that we want to assess for an irritation potential. So barrier function, and they do have to have these lower and upper acceptance limits listed, and this information is also provided in a C of A, so a certificate of analysis, when you order a lot of tissue in the lab to ensure that they are meeting their specifications. Another condition that has to be demonstrated is a functional level. So we want to make sure in the manufacture of the tissue that there's consistency. We don't want the tissue to be too thick or too thin because that will bias the results. So that's where we get this viability limit here, where we show, you know, how much tissue has grown, so how much of the, they say viability, but how much of the, you know, the measure of this component is being processed. So this shows good consistency in manufacturing. And again, I put morphology listed here as well. So this is, again, a histological examination of the reconstructed human epidermis. So again, doing the tissue staining to show that there's differentiation in the cells. So that's a background on the actual tissues. So as a company who may pursue this, this is not something you will look at, but I think it's important for you to know this is what us as a laboratory is looking at. This is what we're looking at in the industry from the tissue manufacturers, so that we're having these conversations to make sure as we move forward, we continue to have a robust test system. Okay, so now I'm just going to go over the details of the test. So this is just a summary to kind of give you a bird's eye view of the test. So we use 15 tissues to run the test. So we run everything in triplicate. So we'll have 3 positive control replicates, and then for each extract, so we do in a polar and a non-polar, as Sophie mentioned, we'll have the control and the test articles. The data is considered quantitative because we are reading optical density from the conversion of the MTT into a formazan salt, and we'll talk about that. The turnaround time right now is 4 to 5 weeks, which for those of you who've done intracutaneous reactivity irritation testing, you'll note that that's no different. The reason for this time right now is that we as a laboratory don't have a high enough number of irritation tests coming in to have a standing order of tissues. And it takes 3 weeks for the tissues to grow. So once we get a study in for in vitro irritation, we place our order for the tissue, we wait the 3 weeks, and then we do the testing. So the testing takes about 1 week, and then report out is about, another week. So we're hoping as the industry gets going on this, we get more and more acceptance in the regulatory realm, that we can cut that turnaround time down. And you'll also see the cost is not different, not much difference from the Invivo. It's also because the tissues are costly, but again, we aren't able to bundle and have a cost-saving opportunity there. So we do expect these numbers to change, and most importantly, we expect to have a lower turnaround time when we get a high enough volume test standing tissue orders. Sample requirement is still 2 samples, and that's so we can have the 2 separate extracts and then 30 centimeters squared each. So quite a smaller amount of sample, and all of that will be clear as I go throughout the next slides when we go into the details. So just for the overall summary, it's all the same steps actually as an in vivo study. We have an extraction, we expose to the test system. Here, instead of scoring tissue sites or injection sites, we're looking at cell viability, and then we do analyze the data. So I want to go through each of these steps to highlight how it actually carries out in the in vitro. And I did put down here what MTT stands for, because I really don't want to be pressured to say that, but it's a very long word. It is a dye, and it is used as a common dye in cytotoxicity, but it's important to note that the dye does not make the test method. So MTT is a dye that can show us cell viability. It's the test system that we apply it to that is, drives the actual result. So I just want to highlight that because I've had a lot of questions. Well, if you use MTT, isn't that just a cytotoxicity test? And it's not, because we're applying it to a more complex cell system. Okay, so for our first phase, the extraction, it's just the same as we would do for in vivo and also for many of the other biocomp studies. We're going to extract in a polar and a non-polar solvent. Typical extraction conditions are mostly done at 50 degrees for 72 hours, but of course we can do 37 if that's justified by the nature of the material or use of the device. So then we want to go to exposure. So this is where the test article extract is topically applied to the tissue, and then we have an incubation between 18 and 24 hours. So this is an actual photo from inside our laboratory where we had some tissues out, and you can see, I just want to point out, so the smaller plastic circle is an insert, is what we call a tissue insert, and at the bottom of that is where the tissue is. And the pink around that is cell culture media that kind of feeds this tissue from underneath. Similar, if you think about our skin, you know, it gets its blood supply underneath. So it's very similar in that aspect. So when we do the exposure, we put these inserts in 3 separate 6-well wells in a 6-well culture plate, and then we expose 100 microliters of the test sample extract. And we'll do 3 tissues for the polar and 3 tissues for non-polar. So when I say we need a small amount of sample, this is the reason why that number is so much lower than in the in vivo. We're only using 100 microliters of test samples, so 30 centimeters squared gives us plenty of test sample to run the test and do extracts, or repeat if we need to. After the exposure, then we will do some processes to measure cell viability, and this is where we use the MTT. So first, after exposure, we do place the MTT on the cells and place them in the incubator. So the purpose of the MTT is to be incorporated or changed to a formazan salt. So MTT is typically yellow, and once we expose it to the cells, the mitochondria in the cell will convert that, if the cell is alive and functioning, convert that dye to a formazan salt. So it will come purple. It's the color change that we'll see. So right here in this image, we've already exposed the tissues to the MTT, and right now we're extracting the color out using IPA. So we'll extract those tissues in the IPA, and you can see from here already, we see some cells or some where there's no color change, so there was no viability there, or not to our visible eye, and you can see some where there's some slight patches where the tissue has survived, but still that's going to be pretty low on the viability scale. So then we take this extract and we'll actually plate it for our data analysis where we're going to read it on the spectrophotometer. So we take that extract and we plate it in a 96-well cell culture plate, and we plate each tissue in replicates. So we do that in triplicate again, and we gather those results and run an average. And that's shown here. Here's our percent viability calculation. This is, put in ISO 10993 part 23. And this data chart is exactly what we used for the validation, where we will look at percent viability, we look at the optical density, and then we create this category of is it an irritant or a non-irritant. And 50% is the cutoff. And that's the cutoff that's also in OECD 439. So if you are above that 50%, the device or material is considered a non-irritant. And if you are below that, at or below 50%, it would be considered a category 2 irritant. So one thing that's important to note right now, we do not have the ability in this method to do a category 3 irritation score where we would say it's a mild irritant. I do think that's something that will come down the road as we refine the method, but right now that option is not available through this test method. So some questions that come up, initially is what devices are suitable for in vitro irritation? What can we use in this? What can we test? So solid devices, which can be extracted, of course, just like we would do for our standard other irritation tests. And one thing I just want to clarify is we do not apply the actual device directly to the tissue. So we would not cut portions of the device and place it on the tissue. This method has not been validated for that. If they're solid non-soluble materials. But what about liquids, gels, and pastes that we typically wouldn't extract? So these can actually be added directly to the test system. So this is what this method was developed for. I think about the cosmetic industry who are applying cosmetics, powders, gels, pastes to the test system as well. However, there does need to be some consideration if the extraction and exposure times are relevant for that type of exposure. And so we do have some options. For here at Nelson, we validated this method for chemicals as well. And so we would want to think about, even though these are categorized as medical devices, what type of exposure system makes sense. And so we do have some options, but you'll definitely want to talk to us and talk to our experts running the test so we can make the best judgment on the method that's appropriate. But that's honestly one of the nice things about in vitro is that we can apply these. We think about in the in vivo method where you're injecting these into animals and it's not quite, clinically relevant. It's great to still be able to assess an irritation potential without having the complexity of dealing with injecting this and dosing the animal and also considering animal welfare. So this is a great thing to consider if you have a liquid, gel, or paste medical device. Okay, so we want to step in just thinking about the data review of the method. So just looking a little more closely at the results. So this is a table that came out of 10993 Part 23. So this is where we get our acceptance criteria. So if you are at or below that 50% viability, again, the device is considered an irritant. This applies if it occurs in either one or both extracts. It's the same as in an in vivo study. If we had seen a failure in the nonpolar extract, it's still considered a failure. The point of the two extracts is to just match a range of solubility of whatever we're trying to pull off the device, because we're not targeting a known irritant. We're just trying to, cover two different ranges of solubility. So if it happens in one or both extracts, it's considered an irritant, or I think we're saying a non-irritant in this one. So yeah, if your cell viability is above 50%, you can consider your irritation assessment complete and that you're passing. So I think this is confusing. So I want to clarify that you have to pass in both extracts. I should have taken this statement out. So if you show passing tissue viability above 50% in both extracts, then your assessment is complete. So in the next slide is where we talk about the failures. So if you are below 50% in one or both of the extracts, that is considered an irritant, and the test sample would be classified as an irritant. So we would investigate this very similar to if you had an in vivo failure. I know that there's some retesting options with the in vivo. We can also look at maybe altercations of the test system, extraction conditions. Is there a known irritant in the product that we already knew would be there? Did we expect the result? So you start going into a very typical biocompatibility failure investigation when this happens. We don't want to try to test into compliance. We want to see if we can understand why it happened and if there's a logical approach to making some adjustments to the method. So there's not a cut and dry answer for addressing an irritation failure because it's always device dependent. There are some devices that actually have irritants into the product as part of the function, and so we think about the pros and cons then at that point. We also just want to briefly mention the in vivo irritation tests as well, because they are still an important part of the assay of the standard. And like Sophie mentioned, there are some applications or locations where a device will be used where a clinically relevant irritation site or analysis site should be selected. And right now, we don't have an approved tissue for that, for the in vitro method. I will say they are being developed. I know the tissue manufacturers have several available, They just have not been validated for medical device. So currently our recommendation would be to use an appropriate in vivo model if you need to assess like ocular or buccal irritation. There have been some changes in the standard with the in vivo methods, but nothing that's changing the overall approach. I mean, we've changed some text and some wording, but the actual approach and application of the products is the same as it was before under -10. So I think the next biggest question is the method accepted? Because it's hard to move forward with a test method if you do the testing and you get to your submission and we find that it's been rejected by the agencies. So what I've done is I've collected some feedback that I've heard as part of my ISO membership, and I'd like to just state these are not firm, this is not something that can be said, well, Audrey said that you guys said you would accept this. We've reached out to the countries to ask what their plan of approval is and how it's working. And one thing to keep in mind, this is an international standard that does need to be translated so that it can be followed and understood by agencies all over the globe. So there will be a period of adoption time, but overall, the feeling that we get is this is widely anticipated, and all the agencies are very excited to be on board with this. So I just want to review some of the things that I've heard going around, and again, I just want to clarify this is not like something you can go to the bank with. It's just to give you an overall perception of where the regulatory climate from a global perspective. So from the U.S., we're getting very close. We tried, I say we, but it's actually Dr. Kelly Coleman who's been a great advocate for this method. And he's worked a lot using an MDDT submission with the FDA. This is a method development tool that the FDA has offered. So right now, the last we heard on that submission is that we have answered all of their questions and submitted all the data they have asked for, and right now we are waiting on their official acceptance of the method or further feedback. Initially, the concern was whether the method was sufficient and comparable to the in vivo methods. So we've submitted some additional data and hope that that addresses the FDA's concerns. So we are very hopeful. We've heard very positive things, and they've had great open dialogue with members in the industry. So we're very hopeful for that to be accepted by the FDA very soon. China has stated that they've accepted -23 as soon as it was published, and they knew it was coming. Again, there will be some time needed to translate the document and get this into their YY standards, is what they're labeled as. So we do expect China to be on board. Korea is also, they're expected to accept this. They've not had any disputations. And China and Korea, US, they've all been a part of the discussions on the ISO level and also participated in the round robin, especially China. So, this is not a new method to them. So we do expect Korea to accept this, and they are working on the translation. So for the EU, we've heard from one notified body. This is from Tooth Food, and they did say that they will accept irritation data while they're transitioning the document to an EU harmonized standard. So they'll continue to accept in vivo, but the in vitro is immediately recognized as state-of-the-art science. But there's not an expectation that everybody will have to submit in vitro immediately. I think that's the greatest thing we want to, think about is, we don't need to all run and redo testing. We need to think about if it's appropriate for the device. But if you're stepping into testing now, I would definitely be considering in vitro if you're assessing something on a device or a new device. And Japan has already jumped the gun. So they have this already included in their MHLW guidance, and so in vitro is now preferred for Japan if you are submitting. Again, this is a transition year, so I think it is appropriate to have a conversation with your regulatory agency about whether they will accept in vivo data if it was already conducted previous to 2021. I think it's important to open that dialogue for your submissions. And then just a quick update on 10993-10. So I think there's a little bit of confusion that we have two standards out currently that are talking about irritation, and one has in vitro irritation as a preferred method, and the other, -10, which is still out and considered relevant, has this as an informative section. So right now, as a team, as a working group, ISO working group, we are working on the new revision of -10, which removes all of the references to irritation testing. We do hope that it will be published this year. I know our convener has been very diligent in working with the ISO organization to get that updated. So we do expect -10 to actually update this year, and just know that the biggest change there will be the removal of irritation testing. And then the sensitization test is what's on task next for us to come up with an in vitro method. And we're working on that, and sensitization is a far more complex reaction. And there's a lot of discussion, but there's a lot of great methods out there that we are looking at, and we're trying to work on developing a round robin. So we'll work on assigning a task force next. And so that's just a brief update on -10. And so to wrap us up today, I'd like to turn the time back over to Dr. Michel so she can summarize our discussion today, and then we'll move to questions shortly.
Thank you, Audrey. And indeed, we are reaching the end of our webinar, which will go, of course, with the conclusion. And so I'd like to step back to, what is mentioning ISO 10993-23 and how you need now to assess irritation. So you now have different tools that you can use, for example, that can be to collect physicochemical information on the device, or to evaluate the amount of literature information that you have. And if you already have sufficient information with these two tools to evaluate irritation risk, with a paper exercise, I would say, then you can stop there and you do not need to proceed with testing. If you do not have enough information at this stage, then you will need to proceed to irritation testing. ISO 10993-23 does ask you to consider in vitro irritation first, and you will still be able to consider in vivo irritation tests, but only if needed and if your medical device is coming into contact with special tissue. So I would like to remember everyone that, of course, irritation is an endpoint of evaluation for medical device, but it's actually only one of the endpoints. There can be several different endpoints depending on the classification of your product. But what is important is to see that ISO 10993-23 actually asks you to evaluate for irritation as part of a risk assessment process, which is exactly going hand in hand with what is requested in ISO 10993-1, which also asks you to evaluate the safety of your final product as part of a risk assessment. And this will be done in a biological evaluation plan, in which you're going to evaluate all the amount of data that you have on your product and how you're going to address the safety of your product, and so actually which strategy you're going to develop. This, of course, needs to be performed before you step into testing, and naturally also before you actually evaluate the safety of your product in a biological evaluation report. So on this slide, you can see different articles which are referring to how the method was developed and validated for medical device, and you can certainly consult them if you want to have more information on the in vitro irritation test. If you do need any help on consulting, for example, to understand how you can address the safety of your product or to design the strategy that would be the best adapted to the biological evaluation of your medical device, you can certainly contact us. We have expertise in this, so we can support you in this exercise. And if you also have any need of testing, you can contact us. And for example, we are able to perform already the in vitro irritation test for medical device. It's validated at our laboratories, so we can certainly help you in this. And to finish, I would like to refer to our homepage and sister companies. You can also go to our website and you can find additional information on medical device, like webinars, or white papers, which can also be very interesting for you and for your team. And with this, I would like to close, I would say the content of the presentation itself, and Audrey and me will gladly answer to any question you may have.
Fantastic. Well, thank you both for that thorough and informative presentation. Once again, don't forget this webinar will be archived on mpomag.com for a year. Now, as Dr. Michel mentioned, it's time for questions and answers, and we've gotten quite a few already. The first one, this one came in quite early in the presentation. Can you clarify what is meant by externally communicating medical devices?
Yes, I can take that. So when we talk about externally communicating medical devices, there's examples in 10993 Part 1 to help guide, an understanding of what devices would fit in that category. But those typically are devices that will be inside and outside the patient at the same time. So we think of, colonoscopes or surgical instruments that are held by the surgeon on one side, maybe even still inside, but not really like implanted in the body, right? So then there's a component that's touching the patient and being manipulated by the practitioner as well.
Okay, great. The next question we got, injection molding sometimes require mold release or mold cleansing chemicals. What documentation would you expect in order to declare that there was no processing aids used in the injection molding process if it was an outsourced injection molding process?
Yeah, so if just what we've seen in the past is just a statement from the manufacturer or the provider of the material that there are no aids used in the injection molding process. And if they state that, the expectation is it's just heat in the material. But as you said, that's often not the case. So we would expect them to say, yes, we use injection mold agents and processing agents, and here's a list, if they would be so forthcoming. And I know that that's very difficult to do. That's why the literature review and manufacturing process can be very limiting, and because we're not always able to get that information from suppliers. So it's a good question, and I think I understand the foundation for it, because that can be difficult.
Okay, great. Next question. Why wasn't semi-polar extraction performed as well as the polar and non-polar per ISO 10993-12? Seems like FDA is wanting all three extractions to be performed, but was not part of the in vitro test. The in vitro test summary?
Yeah, so that's a great question. We are seeing a request for the semi-polar solvent, and it's most commonly made for chemistry analysis. So if we're doing extractable, leachable testing according to 10993 Part 18. So when we're doing the biological systems, 10993 Part 12 still does say most commonly to consider polar and non-polar, and that is what's done for all other biocompatibility assays except for cytotoxicity and material-mediated pyrogen tests, where the specific solvents are needed to accommodate the test system. So I don't think it wasn't consciously excluded, but it wasn't even thought to include, because that wouldn't be a standard inclusion for this assay. So the FDA does ask that for chemical testing, though.
Okay. The next question we have, and I think you covered this in that final section, but just to recap it again, do you know if the U.S. is accepting the in vitro method?
Yeah, and I did definitely cover that, but it bears to be repeated that we want to, we're still having those conversations with the FDA. I think we'll try this avenue with the MDDT still, but other avenues I've seen happen, even specifically from medical device manufacturers, is they will ask the FDA to review a standard and come out with their review of it, whether they'll list it in their accepted methods. So I did see that for a standard not too long ago. So I would say if there's a company that's fairly anxious and would like to have the FDA's input and wants to go that avenue and they're in the middle of a submission, you can formally request that as part of your submission.
Okay, fantastic. The next question we got, per ISO 10993-1, are there guidelines for literature review of chemical mixtures?
Yeah, this is a tough question because even the chemistry testing we do, and Sophie could even chime in here if you'd like to, and maybe even correct me if I misspeak, but even the chemistry testing is not appropriate for analyzing mixtures. We don't have a great way to do that. So if the chemistry is unknown about the mixture, then some data on that specific chemical reaction will need to be gathered. And we've seen that before where maybe we're testing something specifically that cures in situ or something like that, so there needs to be a discussion about the chemical reaction as well. So I would say work with a consultant before you decide, a chemistry expert, before you decide to go straight to all the testing to see if there's some more information you can state about the reaction that's expected.
Okay. The next question we got, is a change in sensitization testing expected?
Yeah, so then that's what I did cover at the end as well, is that we're hoping, right now 10993-10 will publish again this year without much change. There's some, I remember there's a little bit of discussion between the laboratories running the sensitization test that we need to clarify some of the language, but as far as anything that would affect, device manufacturers, there is not a change on that side. We are building a task force, working on building that task force so we can move to an in vitro round robin for that. So that will be coming, but that one will take, I imagine, a couple of years before we'll come up with an in vitro method that's been validated.
Okay, we have another one. If the medical device is a biodegradable orthopedic implant, is in vivo mandatory?
That's a great question, and we honestly don't know at this point if you would have to do in vivo. I think the in vitro method is just as applicable for an implant as it is for a surface device because it doesn't discriminate against that application, and the barrier system there is sensitive enough for an implant in the tissue. So I do think that it's appropriate. The question with biodegradables is always we'll need to assess it at multiple states of biodegradation. So I think the in vitro method is a great option because it allows you to do a lot of that side by side at different levels of degradation. If you can pre-degrade, the samples and then assess them, again, sparing a lot of and still protecting the patient, which is the ultimate goal.
Okay. And this one is a follow-up to the earlier injection molding question. Do changes in process parameters such as temperature trigger new testing or reporting if there are no changes to the materials?
They don't. This is a good discussion to have about what is an actual change for a device. So when we do biocompatibility, we are assessing not just the materials, but also the processes. So if there is a change in that process, it should be reviewed to determine if it's significant enough that we expect there to be some more residuals on the actual device. And so it should be evaluated. A lot of times we'll look at the processing though and we'll say, here's all this processing and maybe one small part of that has changed and then it goes through the same cleaning process. And so we'll kind of think about, would we expect it after that cleaning? Is the cleaning process sufficient enough? And then possibly gather some additional data if we think it's needed based on what's changed. Again, if you're adding an irritant or a known irritant, we would want to look at, just thinking about if we can assess if that's been fully removed before it sees the patient.
Okay. Another question, a lot of questions today. Does the in vitro 23 method support product testing with antimicrobials?
Antimicrobials are not specifically mentioned as, how to assess the irritation potential here. With antimicrobials, we often see, and maybe this participant actually has experience with it running into problems and irritation, but our most common difficulty is actually getting cytotoxicity to pass with an antimicrobial included in that. So we haven't specifically addressed that in the standard, but again, if you run the standard assay and you fail and you can identify, it's most likely due to this addition, then you start running some risk analysis addition. Do we need to, is this actually pose a risk to the patient? I mean, we're seeing an irritation in a very sensitive system. Is it used clinically already? You kind of go down this risk assessment path and determine maybe we do some adjustments in the method to account for a clinical exposure as well. So that's a long answer to say it's not included in the standard, but the thought process to address that is included in -1 and also in -23, looking at a risk-based approach.
Okay, another question. Can the in vitro skin irritation be used to replace the intracutaneous reactivity test in vivo? Yes, that is the absolute approach, and it is written in -23 with some explanation. So I know it seems difficult to think about, this as a skin test. Can we look at intracutaneous reactivity, which was, not on the surface of the skin? So what's important to remember is although this is tissue that's grown up with cell differentiation, it does not have the same barrier properties, right, as like what would be on my tissue here. It's still, meaning that it's more sensitive still. So not to the point that we're seeing false positives, but to the point that it can and it is as predictive as the in vivo test. And that list of papers there that Sophie presented has information and data on that, and you can also do further research. We just published another paper this year about that, and that's what we submitted actually to the FDA because that was their concern. So these are great questions. You guys are thinking just like the industry is, and we have worked really hard to answer those. So the answer is yes, it can and does replace the in vivo intracutaneous reactivity method. Okay, another question we have. Can we still continue using the in vivo test based on company history, or can we expect pushback from the authorities? Yeah, that's a really good question. And I think that for now you can continue in vivo, but I do imagine especially, Europe's really been on board with reducing animal testing, which is great. That's where we want to go as an industry. And I have heard the most that, the expectation will be people are moving to this in vitro method. So I think for now, for 2021, you could probably continue in vivo. But after this year and more acceptance, especially when we hear from the FDA, we really won't, it really won't be applicable to go to in vivo first. Okay, well, I appreciate all, fantastic questions, great responses, but unfortunately that is all the time we have for today's presentation. I'd like to thank our presenters, Dr. Sophie Michel and Audrey Turley, for their insights. I'd also like to thank our sponsor, Nelson Labs, for its support. Finally, I'd like to thank you, the viewer, for attending this session. I hope you found it to be a valuable experience.